So Chloe Stevens is a Senior Scientific Officer for Refinement in the Animals in Science Department at RSPCA, which is the largest animal welfare charity in the UK. She works to promote refinements and improvements in welfare for animals used in research and testing, with a particular focus on fishes and other aquatic species. She has studied behavior and welfare in a range of fish species, including zebrafish, guppies, and mollies, and has a PhD from the University of Exeter in stress biology, welfare, and mortality in ornamental fishes. So, Chloe, welcome to the virtual stage, and we are looking forward to your presentation.
Thank you very much, and thank you for inviting me to speak to everyone today. Good afternoon, everyone. Hopefully you can all see my slides, and someone will hopefully let me know if you can't. So as Ord said, my name is Chloe Stevens. I'm one of the Senior Scientific Officers at the RSPCA in the UK in the Animals and Science Department. And if you're not familiar with the RSPCA, we are the largest animal welfare charity in the UK and one of the oldest in the world. And in the Animals in Science department, we work to reduce the impact of science on animals and their welfare. And we do this by working constructively with governments, with academia, with regulators, with industry, and also other NGOs in the UK, EU, and internationally as well. And we do this through two major work streams. Firstly, we work to ensure that systems of ethical review are robust and operate effectively to challenge whether and how animals are used in science in the first place. And our second work stream is to promote the fullest possible implementation of the 3 Rs. And my work today forms part of our work on refinement. So to set the scene, why did we start working on enrichment for zebrafish in the first place? Well, as I'm sure pretty much everyone here is aware, the use of zebrafish in scientific research has seen an incredible increase over the last 30 years or so. This graph, which I imagine many of you will have seen a version of this before, it shows the number of original research articles published each year, which had zebrafish as a keyword in the dark blue bars. But some of you might have already spotted that it also shows the number of research articles published each year containing the keywords zebrafish and housing or husbandry or welfare. And those are in the teal bars. And if you're struggling to see those teal bars, well, here they are. So what this suggested to us was that despite the rapid increase in the use of zebrafish, there wasn't a similar increase in researching how to provide zebrafish in the lab with the best possible housing and husbandry conditions. And this in turn suggested that there was probably scope to look at how we might improve zebrafish welfare. And so we decided to focus on enrichment as a potential way to do this. And I imagine that most of you are pretty familiar with what enrichment is, but just in case, a simple definition of enrichment that we can use and that we have used for the purposes of our work is modifications that are made to the environment of captive animals with the aim of improving their welfare. And there are some examples on the slide of enrichment for several other common laboratory species. And for zebrafish, modifications that might be used, or at least might be thought of when people discuss enrichment, include live food, social housing, and more recently, things like images of gravel underneath tanks, as well as real gravel or real or artificial plants and other tank ornaments or objects. And when we first started looking at enrichment for zebrafish, it seemed that enrichment use was not particularly widespread. So a survey that was conducted by the UK's National Centre for the 3 Rs in 2017, which looked at all aspects of zebrafish care and use, found that only about 25% of the respondents were using enrichment in zebrafish tanks, although most were using live food. Indeed, many zebrafish labs may look similar to the ones on this photo, with tanks that are small and fairly featureless and don't appear to be particularly enriched. But if we have a look at a picture of some wild zebrafish habitat, this is some wild zebrafish habitat from Nepal, you can see that it looks like this. It's got lots of plant life, there will be water flow, there will be substrate at the bottom of the water column, there'll be reductions in visibility, there'll be other animals in the water, there'll be seasonal and diurnal changes. So what you've got here is an environment that is both highly complex and also highly dynamic. And if we compare this complex and dynamic environment to the relatively barren tanks that we tend to keep laboratory zebrafish in, we can see how these tanks are likely to place restrictions on how zebrafish express their natural behavior, and this is consequently likely to cause problems for welfare. And enrichment is a way that we can try and reduce those negative effects of laboratory housing. So why isn't enrichment use more widespread? Well, this is a study that was conducted in 2019 by a group of social scientists at the University of Oxford. And what they did was interview technologists, facility managers, and others who were connected to the use of zebrafish in labs. And they identified enrichment for zebrafish as a hot topic. But lots of interviewees reported little enrichment being used in their facilities. And one of the major reasons they identified as to why enrichment wasn't more widespread was a concern that there just isn't enough evidence in favour of using enrichment for zebrafish, or to say what kind of enrichment zebrafish want. So here's a couple of examples from interviews that they did as part of this study. The first quote on the slide there: "I think probably the biggest constraint is just actually the lack of good data and scientific knowledge about what an appropriate environment for the zebrafish might be." And that second one: "We can't make that much more progress, I personally don't feel, unless we can really say this is what is good for them in terms of environmental enrichment." But it certainly seems that enrichment might be worth looking at to improve welfare. Here are some of the benefits of enrichment that have been observed across all sorts of different species and different taxa. And you can see there's physiological impacts, like impacts on growth rate, immune system function, and neural development. But there's also behavioural effects, such as reductions in anxiety and aggression and neophobia. Now, sometimes in discussions with people, I hear people express doubts over how applicable those sorts of results might be to fishes, and particularly to zebrafish. So it may surprise some of you to know that actually almost all of these benefits have specifically been demonstrated in zebrafish. And I'll talk about some of the papers that have led to these sorts of results in a bit more detail shortly. But just to summarize the more kind of general attitude of why we should use enrichment then, well, first of all, I would argue that we have an ethical or a moral responsibility to try and provide the animals in our care with the best possible welfare we can. Most of us will also have a legal or a regulatory responsibility to improve welfare, and what that is will depend somewhat on where you are in the world and the laws or regulations that you're working under. But many of these relating to the use of animals in science do include a responsibility to try and improve welfare. And the final reason is encapsulated by the old adage, which I'm sure that everyone's very now familiar with, that happy animals make good science. Or perhaps it's better to say good welfare makes good science, because it might be difficult to say whether the animals are happy per se, but we know that animals need to have normal physiology and behavior for us to get good, valid, robust data from them. And that means prioritizing welfare for its benefits, both for the animals and for the science we're doing with them. So what impacts does enrichment actually have on zebrafish then? How is it affecting their physiology, their behavior, and their welfare? So the first part of our project on enrichment involved conducting a literature review. So we combed the literature for every paper we could find on enrichment. And for each paper, we summarised the enrichment strategy that was studied, the welfare parameters that were used to assess the enrichment, and the effects the enrichment had on the zebrafish. And those results are all summarised in these easy to read tables, and the paper is fully open access. So do feel free to take a look if you're interested. And as I say, if you don't have time to read the whole paper, the tables summarise all of the studies that we looked at. And it's available through the tiny URL link on the slide, which will also be up on the slide at the end as well if you want to look it up. So I'm not going to go through all of the papers that we looked at, but I'd just like to highlight a few key results that we looked at in our paper. And I'm going to do this using Bloom-Smith's 5 categories of enrichment: physical, social, sensory, dietary, and occupational enrichment. So let's start with physical enrichment. So this is the category that is probably best studied for zebrafish, and it refers to physical objects or items in the zebrafish tank. And that might include things like plastic or real plants, rocks or pipes for shelter, or substrates like gravel or sand. And physical enrichment can increase the complexity of the animal's environment, as well as doing things like providing refuges or shelters from animals or from lab staff as well. So one of the key questions for any form of enrichment is, do the animals actually want that enrichment in the first place? In other words, do they show a preference for having that form of enrichment over not having it? And that is exactly what this first study did. This is probably one of the best known papers on the topic. It's by Paul Schroeder, and it was from the lab of Lynn Snedden, who just gave us a wonderful talk. And so what the authors did was they housed zebrafish in tanks with 2 chambers which the fish could swim freely between, and each chamber contained a different condition. And they looked at percentage occupancy. how much time were the fish spending in the side of the tank containing the enrichment compared to the barren side? And they found that for several forms of enrichment, including but not limited to gravel, sand, and a floating plant, the fish showed a really strong preference. They spent much more time occupying the side of the tank containing the enrichment over the barren side. And as I say, this is probably one of the best known studies on this particular topic, but it's not actually the only one. There are several others that have found similar results, that zebrafish prefer structured environments, that is, environments that contain physical enrichment. So this is a result that has been replicated. As well as looking at preferences, we also want to know what the effects of enrichment are on zebrafish physiology and behaviour when they're housed with that form of enrichment. So that's what this next study did. So this was a group from the University of Exeter, and what the authors did was house zebrafish larvae either in a tank containing gravel and a plant or in a barren tank. And at 30 days post-fertilisation, so when the larvae were about a month old, they measured survivorship. And what you can hopefully see from the graph on the right-hand side of the slide is that larvae which had been raised in the enriched tanks had much higher survival rates, over 80%, than those raised in the barren tanks, which was about 55%. Also, after a couple more months, the zebrafish were tested in the novel tank test, which is a validated and commonly used test for measuring anxiety-like behaviour in zebrafish. And they found that anxiety was lower in fish raised in the enriched tanks as well. So enrichment in this case was reducing anxiety and improving survival. And once again, there are several other studies that have found similar welfare benefits from being housed with enrichment, such as lower stress after a stressful event like being chased with a net, lower anxiety, including after unpredictable chronic stress, and also improvements in learning, which may suggest better cognition and potentially better brain development as well. One of the last pieces of work on physical enrichment that I want to highlight for you is one that's particularly interesting to me. This is a PhD thesis by Michelle Lavray from the University of Guelph. And one of the first things she did was to study zebrafish preferences for enrichment. But what she did was she did this so that they could be arranged along a scale of preference. So she was able to show that zebrafish preferred plants to an empty tank, gravel to plants, plants and gravel over gravel alone, and more plants and gravel over fewer plants and gravel. What she then did was she housed fish either in their most preferred enriched conditions or in barren housing, similar to sort of standard laboratory housing. And she looked at probability of survival and found that probability of survival was higher for fish housed in enriched tanks. In other words, fish housed with enrichment were living longer, which I would say is pretty compelling evidence that physical enrichment is very beneficial for zebrafish. Now, what about aggression? And the reason I want to mention aggression specifically is because I sometimes hear people express a concern that putting objects into the zebrafish tank is going to give the fish something to be territorial and fight over. And indeed, some studies have found that enrichment can be associated with increased aggression. But these studies aren't necessarily telling us the whole story. So here's an example of a study where the researchers put pairs of zebrafish into tanks either with a plastic plant, which is the red line, or without a plastic plant, which is the blue line. And they looked at wound-free survival. How many pairs survived without any fight wounds over 10 days? And what they found was that pairs housed with a plant had 100% survival at the end of the study, but those housed without the plant had only 55% wound-free survival after 10 days. So enrichment here was reducing aggression-related morbidity and mortality. And again, this is not the only study that have found that enrichment can actually reduce aggression, and it could in fact be really useful for pair-housed zebrafish. And in fact, some of these studies have looked at aggression in zebrafish housed in groups as well and found that enrichment can reduce aggression. So it's likely that there are more factors at play here than just the simple presence or absence of enrichment. This may be due to differences in zebrafish strain or something like stocking density. But what I would suggest is it indicates that it is worth providing our fish with physical enrichment, but perhaps just monitoring them a bit more frequently when enrichment is first introduced to check how aggression is affected. And as a side note to this, I often think that if aggression is increased by the provision of enrichment, that suggests that if the fish or if the animals are willing to fight over it, that suggests that they may want access to that resource. So rather than simply removing the enrichment, we need to think about ways that we can provide it and what else we can change to reduce that aggression. The next type of enrichment I want to talk about is social enrichment, and that's providing animals with other individuals to create an enriched social environment. Now, presumably, I don't have to convince anyone that zebrafish are a social species. We know that they live in groups in the wild, and we also know that given a choice in the lab, they prefer to be near other fish. They prefer zebrafish over other species if they're allowed to choose, and they generally prefer larger groups over smaller groups as well. Is that the whole story, though? Well, there are certainly studies that have found that being in groups instead of being isolated is associated with lower cortisol, so lower stress and lower anxiety. But there are also studies that have found the opposite. And there's also some that I haven't listed here that have found no effect on cortisol or anxiety. So what's going on here? Well, it may be that there are other factors at play. Perhaps what matters is not the presence of a group or the absence of a group, but how that group is composed. For example, some studies have found that single sex housing can have improvements on several parameters that are associated with welfare. And although I'm not recommending single sex housing for zebrafish because that comes with a host of other implications that may affect welfare, what this does support is the idea that group composition matters to zebrafish. We also know that zebrafish recognize familiar individuals and interact differently with them. So maybe being with a group of familiar individuals is more relevant for welfare than being in a group of unfamiliar individuals. There could also be factors at play, as I've touched on before, like the stocking density used in these studies, perhaps the welfare parameters that were actually used in the studies and how they were applied, or a combination of these things or something else entirely. It's not clear. But the point I want to make here is that what I would conclude from all of this is that while zebrafish should be housed in groups, we do need to be aware that social dynamics are complex, and therefore so is social housing as a form of enrichment. So we cannot simply say that fish housed in groups are in an enriched environment. Now, there may be some of you who have been thinking all through this section, well, of course, social housing is just a basic need for zebrafish, not an enrichment. But I have heard some people claim that social housing is a form of enrichment they apply to they provide for their zebrafish. And I would say that we cannot necessarily conclude that. Providing company of any sort is just fulfilling a need rather than providing enrichment. So if social housing is the only form of enrichment you're providing for your zebrafish, I would strongly encourage you to think about how else you might be able to introduce enrichment. And as a broader point, we do need more research into factors that may help us understand what a socially enriched environment looks like for a zebrafish, because what those results on that previous slide also show us is that social environment does matter. So we may indeed be able to provide zebrafish with a socially enriched environment, but we probably don't know enough yet about how to do that. So now I'm going to move on to sensory enrichment. So these are enrichments that might involve visual or auditory or olfactory stimulation. And I think this is quite an exciting area of potential enrichments because they often involve things that don't necessarily require putting something into the tank. So do we have evidence that zebrafish respond to these kinds of stimuli? Well, yes, we do. And to demonstrate this, I'm going to go back to the first study that I showed you, which was this one with the choice tanks. And if you look at this image on the bottom right of the slide, you can see an example. So in this condition, the zebrafish were tested for their preference of an image of gravel over a barren tank. And just as with the physical enrichment, you can see that the zebrafish much preferred being on the enriched side of the tank. So this shows that just the presence of a picture outside the tank was enough for zebrafish to express this preference about the kind of conditions they wanted. And in fact, if I put the graph for real gravel back up here, you can see that the strength of preference, the amount of time spent occupying that side, is almost as strong for the gravel image as it is for the real gravel. Similarly to some of the to this study, there are other studies that have looked at the effects of visual enrichment. So this one by Woodward and colleagues found that an image of a sort of seascape on the walls of the tank led to higher egg production, which may have been a result of lower stress and better welfare, although that link is perhaps a little bit more tenuous. And visual enrichments could also include things like tank color. For example, this is a study that found that fish had lower anxiety in blue or black tanks instead of white or transparent tanks. And a form of enrichment that's been proposed, but unfortunately not directly tested as far as I'm aware, is the inclusion of dawn and dusk phases in the lighting cycles used in the lab. And the idea is that this may reduce stress caused by the startle response that can be seen in zebrafish when they're exposed to sudden changes in light levels. So as I say, some exciting potential ideas there, although admittedly an area that has not been as well researched as physical enrichment, but something that I hope we will see more results coming out about in the future. But of course, sensory enrichment doesn't just have to be visual. It can be via other modalities as well, including sound. So in this study, the authors housed zebrafish either in tanks that were exposed to the ambient noise of the lab or exposed to classical music. And they tested their fish for anxiety in the novel tank test that I've mentioned already. So they placed the fish in a novel tank and looked at their behavior. And they found that for many of the measures used in this test, the fish that were exposed to music showed behaviors that are associated with lower anxiety levels. So the first graph here is showing you that the fish exposed to music were spending more time in the top of the tank. The second shows that they were traveling further in the top of the tank. And the third shows that they spent more time being mobile in the top of the tank. And as I say, these are all measures associated with lower anxiety. And yet again, this is not the only study that found effects of auditory enrichment. It was a result that was replicated in another study a couple of years later. So the next type of enrichment I want to talk about is dietary enrichment. Now, in this case, because laboratory animals should ideally be provided with a complete diet, I'm not referring to anything that has any sort of nutritional benefit, but rather enrichments that can improve welfare through some sort of benefit external to the animal, such as providing variety. So for zebrafish, this would most likely be through the provision of live food. Now, live food is usually suggested to provide opportunities for displaying natural behaviours and perhaps to alleviate boredom. But despite having looked many times for studies that demonstrate a welfare benefit of live food, I haven't been able to find any empirical evidence for this that I know of. So if you know of a study that has looked at this, please let me know. And this isn't to say that live food isn't enriching. I think any of us who have provided fish with live food in the lab have probably seen the difference in behavior in the fish. And I think it probably is enriching and I would encourage its use. But I think this is something particularly interesting to think about because live food is commonly described as an enrichment and commonly used as an enrichment for these sorts of behavioral reasons. And yet enrichments for which there's much more empirical evidence, like physical enrichments, are much less widely adopted. And if we go back to that reasoning that I talked about at the beginning, that there's not enough evidence, I think that's a really interesting comparison. And I think it was worth reflecting on how much evidence we're actually willing to accept or we feel we need to make a change when it's a change that maybe it might benefit the animals but might be a little bit difficult to implement. But that's all I'm going to say on that topic. So the final category of enrichment I want to talk about is occupational enrichment. So this refers to enrichments that give animals something to do, essentially. So in other species, it might refer to things like toys or puzzle feeders. And it's a bit trickier to think about how we might provide something like that for a zebrafish, but there may be ways to provide it. And one way that might work is by giving the fish water flow to swim against. So in this paper by Kirsty Di Pasquale and colleagues, the zebrafish were placed in a multi-chambered choice tank, and they were given the choice of a barren compartment that they've called plain here, one that contained water flow, which they've called swimming only, one that contained plants and substrate that they've called enriched only, and one that contained both water flow and plants and substrate, which is the enriched plus swimming condition at the top. So on this graph that shows the results, bars above the line show a preference or more time spent in that condition, and bars below the line show avoidance or less time spent in that condition. And bars on the line show sort of a degree of neutrality. So what you can hopefully see is that fish were fairly neutral towards the central arena. They avoided the barren tank and tended to avoid the condition with water flow only. They tended to spend time in the condition with physical enrichment, but the condition that was by far the most preferred was the one that was enriched plus swimming, the one that combined physical structures with water flow. So this is really interesting, the fact that they were not keen on the water flow only condition, but they were when it was combined with physical enrichment. It suggests to me that water flow might be valuable to zebrafish and something they want access to, but perhaps there are caveats here. Perhaps they only want access to water flow, maybe because the physical structures are in place to break up the flow. Maybe it introduces a degree of choice over how much the zebrafish react with that water flow. It's not clear, but I think it's something that is worth bearing in mind, that water flow may be beneficial for zebrafish welfare in some conditions. And access to water flow has also been shown to have impacts on behaviour. So this experiment, what they did was they placed fish in a swim tunnel each day, and they found that these fish that they called exercised fish were less anxious than fish that weren't put in the swim tunnel, that were unexercised. And from some other papers, there's also evidence of things like improvements in learning and cognition in exercised fish, which might lead to a better ability to cope with stress, lower latency to feed after a disturbance, which might indicate that the fish were less stressed and potentially better able to cope with stresses. And there are also some papers that have shown some of the physical benefits from exercise that you might expect from exercise similar to humans, things like lower oxygen consumption, increased bone volume and mineralization, and these may also point to better overall health and in turn better welfare as well. So those studies have hopefully convinced you that there's a wide range of evidence that shows that enrichment is beneficial for laboratory zebrafish. But if that's the case, why isn't everyone using loads of it? Well, there are, of course, various challenges that people might face when trying to implement more enrichment for zebrafish, and we wanted to better understand what these might be. So we conducted an international survey on the use of enrichment in zebrafish facilities, and we got about 100 responses. And if there's anyone in the audience who completed that survey for us, then thank you very much. So we asked participants about what kind of challenges they faced when trying to introduce more enrichment, and you can see how many people responded with different challenges on the slide. So top there is concerns over effects on water quality, but also things like resistance from researchers or other staff. So we think that that might relate to some of those points we were talking about at the beginning about not being convinced about the evidence. We did include not being convinced of the evidence as a potential option, and people did say that that was a factor of one of the challenges that they faced. But we also thought that respondents who were more likely to fill in this survey might be a bit more open to filling in the survey. So it might be that they themselves were convinced, but they were working with people who weren't convinced. But what you can see that there are a range of challenges here, and we did also include the option for people to enter new challenges into the survey. So they didn't just have to answer with challenges that we had previously suggested. We also asked respondents to identify what the biggest barrier was, and you can see that there's quite a lot of variation in the answers. So there's a few associated with the use of physical enrichment, such as the water quality or difficulties in conducting health checks. There are some that seem likely to relate to this lack of awareness of the evidence or not being convinced of the evidence or resistance from other members of staff. And there are some, of course, that are very practical, like cost or not enough staff time, and probably ones that most people can relate to. I'd just like to say that I'm not going to go through too many other results of this survey. We are running running this survey again for people who work with zebrafish in Switzerland, and I'll touch on that again at the end. But if you would like to be involved in that version of the survey, then do please get in touch and we can send you a link. With all of that in mind, the evidence, the challenges, and so on, I wanted to finish with a few thoughts about how we might approach implementing an enrichment program. So my general thoughts here are that one of the key things to make sure of when introducing any new enrichment is that whatever you do, whatever you choose, it is important to make sure that the enrichment is actually having the welfare benefits you're aiming for. Sometimes enrichment strategies just don't work like we expect them to, and at best this can be a waste of time, money and resources, but at worst it can actually be detrimental to animal welfare. So that usually means starting small and properly evaluating the impact of whatever enrichment you want to try. So to start, what might you want to choose to try? So you've seen there are a range of different options, which is great, but that can also make it really difficult to choose, especially as many papers, such as the ones that we've looked at today, tend to compare enriched to non-enriched conditions rather than comparing amongst different types. So we don't necessarily have an obvious best enrichment strategy, but a couple of questions that you might want to ask to begin with are, what outcomes are you hoping to see? Do you want to see a decrease in behaviours like aggression? Do you want to lower the fish's anxiety or stress levels? Or do you simply want to create an environment that the fish have shown they prefer? Similarly, you may want to think about your target group of fish and whether they have any particular considerations that you need to take into account. Are you working with a more aggressive strain? Are you looking to provide enrichment for breeding pairs or for stock tanks? What about fish that might temporarily have to be placed in isolation, such as when they're being genotyped? And asking these sorts of questions can help guide your choices. We can also be guided when it comes to choosing approaches by looking amongst the results of some of the papers that I showed you. So that first paper looking at the overall degree of preference shown, we might be able to conclude from these results that gravel and a gravel image may have reasonably similar benefits, or at least are reasonably similarly preferred by zebrafish. But when comparing submerged plants to floating plants, we might conclude that floating plants are better to try than submerged plants. And we can also look at work like the thesis by Michelle Lavery that I mentioned earlier as well, in which she demonstrated that scale of preference. So looking at trying gravel or a gravel image may be more beneficial for welfare over artificial plants, but also that more enrichment is better than less enrichment. And that really highlights the importance of trying something rather than trying nothing. But it's also important to remember that there are lots of other types of enrichment. It doesn't just have to be things that you put into tanks. And some of the non-physical options that may be worth trying might be images on the base or walls of the tank, extra dietary enrichment, auditory enrichment, dawn or dusk phases in lighting cycles, visual or olfactory contact for singly housed zebrafish, or even combinations of these things, although it's probably best to try one thing and then introduce the next thing, and make sure you're evaluating these things as you introduce each intervention. So once you've chosen the form of enrichment of interest, how do we know that it's working? So as you'll have seen from the papers I've discussed so far, there's a wide variety of approaches that you can use to assess the impact of enrichment on welfare, and which you choose will depend on things like how much time you have available and perhaps the type of data that you already collect in the lab. So you may want to use behavioral approaches like studying differences in behavior using an ethogram. You may want to conduct a preference test, as we've seen in various examples of. In some species, another approach that is sometimes used is motivation tests that look at how much effort an animal is willing to expend to access a resource. And that's perhaps harder to set up with zebrafish, although it's not impossible. Another behavioral approach is to use a validated test of anxiety, like the light dark test or the novel tank test that a few of the papers that we've looked at today have used. And you could also look at physiological measures of stress, like cortisol release rates or opercular beat rates. But what I'd say is that many of these measures, particularly the two that I just mentioned, are more suited to acute or short-term stress rather than chronic stress, and that also interpretation of any single physiological measure can be very tricky, as parameters like cortisol or opercular beat rate may increase or decrease for reasons other than stress. So if you're interested in collecting physiological data, I'd strongly suggest also collecting some behavioral data and or potentially multiple welfare parameters to help interpret those results. So of course, once you've conducted your trial, and you have your results, you can then decide if it's something that you want to implement on a larger scale. But whatever your results, I want to really encourage you to share them in some way so that we can benefit from that knowledge, even if it's for an enrichment type that already has evidence in the literature. Remembering, of course, that not being convinced of the evidence still seems to be a significant factor when it comes to uptake of enrichment across the sector. So making those results available is really important, whether that's through publications, posters, conference presentations, or similar, or recognizing that some of those things are perhaps easier said than done. I'd like to highlight a couple of ways that you might be able to do that otherwise. So another way you might be able to share those findings is by adding them to online repositories. So for example, Noracopa, the Norway's 3Rs Centre, has a refinement wiki, and you can add your findings to that, and that can be a good way to share your results without having to go through the publication process or applying for conference presentations. You may also be familiar with the website protocols.io. So this is aimed at sharing methods to help promote reproducibility, but some facilities are using it to share their facility environmental parameters, which I think is absolutely fantastic practice. And you could include your enrichment program in that kind of document as well. So utilizing these sorts of repositories that are open access can be a really great way to share your strategies. So if you would like some support for how you might go about doing a study to evaluate enrichment, I encourage you to take a look at the resource on the slide. So this was jointly produced with the UK's National Centre for the 3Rs and the Institute of Animal Technologists and the RSPCA. It's aimed primarily at technicians, so it's written in accessible language, and it focuses on the use of behavioural studies to assess welfare, but it should be useful for anyone who wants to conduct this kind of study, and it provides guidance on every step from planning to sharing your findings. For example, this is a table from the resource that gives examples of the types of enrichments you might want to try for zebrafish in different housing conditions. And it also includes things like example ethograms for zebrafish, if that's the approach that you want to choose for evaluating the enrichment. So the ethogram I had up a couple of slides ago is available via this resource. So what about those challenges that I mentioned earlier then? Well, a lot of them are difficult to deal with, and I certainly don't want to minimize that. But I just want to give a few suggestions of how you might be able to begin to address those sorts of potential barriers. So hopefully all of you are now convinced of the evidence for enrichment, but if you're not, this is where running a study or a trial using that resource is really helpful, because you can see for yourself whether it's going to work in your facility. And of course, you can take a closer look at some of the resources I've highlighted for you today, such as our paper, and also some of the other RSPCA resources as well. And of course, feel free to get in touch if you'd like help or have questions about any of these. Now, if you're completely convinced but you're working with others who aren't, you can, of course, again, share our paper and other relevant RSPCA resources with them. Something else you may be able to do, and this depends on which country you're in, but if you're in a country that has an animal welfare ethical review body or an animal welfare body or an IACUC, I don't know where everyone in the audience is currently, but if there is a body like that, these are bodies that have responsibilities to help promote animal welfare, and going to those bodies to get buy-in and support can be really useful for getting the support that you need to implement some sort of enrichment program. If you have concerns that are about things like the impact on water quality or the view of the fish, or other reasons that are associated with putting objects into the tank, you can always ask the supplier of any enrichment items to provide a certificate of analysis to show that the item has been tested and there's not going to be any chemical leaching and it's safe to autoclave. But you can also look at trying non-physical options like some of the ones that I highlighted earlier. If cost is a barrier, I again would like to encourage you to consider non-physical options like auditory enrichment, or try a bit of DIY. So one respondent in our survey, for example, mentioned using cut-up mosquito netting because that's autoclavable and kind of mimics artificial plants. Similarly, you don't have to buy branded runners that have images of gravel on them. You may be able to make your own, and that can be as simple as printing and laminating some images if that will fit with the kind of tank setup you have. And as I say, if you're simply not sure where to begin or what to try, I would say that some of the things you can do are look at going for what you might think of as the easy wins. So putting plants in tanks, perhaps just for single or pair housed zebrafish, if you have temporarily singly housed or pair housed zebrafish, or putting gravel images under tanks as well. And again, you can, of course, look at our evaluating enrichment resource. So the key message really for me is that there is good evidence that enrichment can improve the welfare of laboratory zebrafish. And even when it's not clear exactly what the best strategy is, we know that it is possible. So we have a clear goal to continue working towards and aiming for. And so anyone working with zebrafish that aren't being provided with enrichment, I would really encourage you to think about ways that you may be able to start to implement this. That said, enrichment should be trialed before implementing it across the whole facility. And my final message is to please try and publish or otherwise disseminate the results of the enrichment trials. So with my last slide, I just want to say thank you very much for listening. I want to highlight a few of the resources from the RSPCA that I've mentioned on zebrafish refinement. You can also see our website and Twitter handle on the slide, as well as the link to sign up to our newsletter if you want to hear about more resources or events that we run. There's the link there to the enrichment review. And as I mentioned earlier, we will be running a version of our enrichment survey for Switzerland. So if you would like to take part in that, then do please get in touch and we'd be very happy to provide you with some more details. Thank you all very much for listening.
Thank you so much, Chloe, for this really enlightening talk on enrichment. So to the audience, feel free to write down your question in the chat or directly in the Q&A. You can also raise your hand if you would like to ask your question directly to Chloe.
Thank you very much, Chloe, for this talk. It was very comprehensive. And I also think that one of the main topics is sharing of experience. When I remember the discussion about non-aversive handling in mice, it seemed to really take momentum when there were resources available, experiences available from technicians, how they work daily with that challenge. And yeah, let's see what will be developed for the zebrafish. We have one question in from the audience. If the gravel image has been compared with other enrichments that are not only visual, like actual gravel plants or other hiding places, and where does it fit in the zebrafish preference scale?
So as I said, the only study that I know of that has directly compared enrichments along a scale of preference was that study from the thesis by Michelle Lavery, and I don't believe that they actually included a gravel image in that particular scale of preference. The study from Lynn Snedden's lab, which did look at the gravel image, compared a variety of different conditions, but I don't recall that the gravel image was compared directly with gravel or other hiding places. So it's more about looking at the degree of preference that the fish showed for that condition, how much they wanted that particular condition, and comparing amongst the other conditions that were shown in that study.
Okay, thank you. Then another question, is there data on the influence of environmental enrichment on recovery, better recovery after invasive surgery in fish? And or is there any data on whether there is a real risk of poor hygienic conditions?
So yes, I believe I have seen a study that is on recovery, or included recovery from a stressful procedure. And I think it is in our review, but I'm afraid I just can't remember the details off the top of my head. So I'm afraid I would have to go and look that up for you. But do feel free to have a look at our paper and see if that's in there. I'm sorry, I can't remember. Sorry, what was the second part of the question?
And the second part is there data that there is a real risk of poorer hygienic conditions when artificial plants or other enrichment are introduced in a tank?
Yes. So one of the papers that I showed, which was mainly looking at aggression, did also do some water samples. So they included 3 different physical enrichment structures. So one was a plastic lily pad that floats on top of the tank. One was a sort of plastic tent that goes into the tank. And one was a sort of more kind of traditional artificial plant. And they did some water sampling and found that there was no difference in there's no detriment to water quality over the course of the study. That said, I believe the study length was only about a week. So I think it's something that does need to be looked into more. And as I say, I would always encourage people to trial enrichments when they introduce them. And so if you're looking at trialing a physical enrichment, that might be one of the things that would be important to look at.
Okay, thank you very much for answering.